Next up, we have Amy VanZanen, who will be talking about her research project with, oh boy, Amy, with Steve Ruberg and Craig Stowe. My eyes lost track of where I was. So Amy, you are up and should be able to share your screen now.
Awesome. Thank you, Mary. Yes, and thank you everyone else for being here today. I'm really excited to talk about my work this summer with Steve and Craig on episodic hypoxia as measured by the Saginaw Bay buoy. So before I get started, I want to make sure to thank not only my mentors, Steve and Craig, for their guidance and patience and dedication to my learning and growth, but I also want to make sure to thank Kyle Beadle, Steve Constant, Ron Muzzy on the OSAT team at NOAA GLERL, and Russ Miller at Sigler, without whom there would be no data from the Saginaw Bay buoy for me to analyze. So a huge thanks to their work in doing that. And I also want to thank Dr. Ashley Elgin for the information she provided on benthic communities. And of course, a huge thanks to everyone who presented to a summer fellow Lunch and Learn. We really appreciate your commitment to students and their growth. So many, many thanks. And I also want to spend a little time at the front to touch briefly on where this buoy is situated in the broader Great Lakes management landscape. So this buoy is on Saginaw Bay in Lake Huron, which is a body of water that provides a lot of economic and ecosystem services. And it's also not known for being the cleanest part of the Great Lakes region. The bay, together with the Saginaw River, are listed as an area of concern under Annex 1 of the Great Lakes Water Quality Agreement. And that means that they have what the agreement terms beneficial use impairments. And in the case of Saginaw River and Bay, those include degradation of benthos and of fish and wildlife populations. And hypoxia is something that might be contributing to that degradation. So as background, hypoxia occurs when concentrations of dissolved oxygen in the water, typically in the bottom part of the water column, are dropping below a designated threshold, which I'll talk about in a bit. And because wildlife require oxygen for respiration, like the rest of us, low dissolved oxygen levels can impact measures of organism health, which is why this is important to study in the context of Saginaw Bay being an area of concern. Currently, we have 3 main sources of GLERL data that are collected on the bay. These include the benthic survey that takes place as part of the Cooperative Science and Monitoring Initiative, and that takes place every 5 years. Last one was this year, actually, and before that, it was 2017. There's also CTD sampling that takes place at 4 locations in Inner Saginaw Bay, and that collects data, conductivity, temperature, depth, among other parameters. And then we have, of course, data that's collected by the Saginaw Bay buoy off Augrey, Michigan, and that's part of the Real-Time Coastal Observation Network, RECON, and it's been there since 2010, thanks largely to funding from the Great Lakes Restoration Initiative, as well as some GLERL funding and support from the Great Lakes Observing System. So we're really glad to have that buoy out there. It provides high resolution data on a number of things. So you'll see me discuss a few of those today, most principally dissolved oxygen. But there's also chlorophyll sensors and pH sensors. So there's a lot of data available from this buoy that's available to anyone who is interested. You can find that online, available to the public, excuse me. So talking a little bit more about this data so when we're looking at this data that's being collected by the buoy, and it's located in the inner part of Saginaw Bay, that's between 10 meters and 14 meters deep, whereas the rest of the inner bay is relatively shallow, and so that provides some differences in hydrodynamics that are at play. And our study this summer looked at data that's between the buoy's initial placement in the year 2010 all the way to 2021, with the exception of some of the data from 2019 because the dissolved oxygen sensor failed for much of that season. The seasons that we looked at run from May to October, roughly. Some years are definitely more complete than others. And some sensors have been added or swapped in over the years. So not all of the parameters that we looked at were collected across all years, but most of the major ones that we are concerned with were, which is good news. So this leads us to sort of our two lines of inquiry. We knew before this, or I should say Steve and Craig already knew before this, based on looking at the online plots, that there have been instances of hypoxia measured by this buoy. But what we didn't know, and what we needed to do, was look at a longer term assessment of some of those trends in hypoxia. So what is the duration, intensity, and frequency of hypoxia that's measured by the buoy, and what might be some of the hydrological dynamics that are contributing to or resolving instances of hypoxia. So those are our two main lines of inquiry this summer. And this is a first look at the dissolved oxygen over time by year. As I mentioned, hypoxia is dissolved oxygen going below a designated threshold, and we chose to use 4 milligrams per liter and 2 milligrams per liter of dissolved oxygen as our thresholds for what we consider hypoxia. And the orange line there represents a threshold of 4 milligrams per liter, and the red line represents a threshold of 2 milligrams per liter. And a couple things that might jump out at you as you look at this is that there appear to be some instances of prolonged hypoxia or prolonged experiences below 4 milligrams and the 2 milligram threshold. So I'll zoom in on a year here so you can see the scale a little bit better. And if you look at the bottom of the graph, you'll notice that the scale is in weeks. And then you can sort of tell from that there's these instances of hypoxia where it's going below 4 and then again 2 for up to even a week at a time, which is a fairly long period of time for water to be at that level. The other thing that we started to notice as we looked at this is that it looks like hypoxia might be more common in later years in this time series as opposed to earlier years. I'll mention that this is a relatively short time series. It's only 11 years worth of data, so it would definitely be worth coming back to this again in 5 to 10 years to see what are some of those longer term trends, even longer than this study covered. So in those earlier years, which we considered 2010 through 2015, you can kind of tell that DO rarely dips below that 2 milligram per liter threshold, that red line there. And 2014 is the first year where it gets below 2 milligrams per liter for any sustained period. Whereas 2016 onward, that is something that happens at least once a year. And in earlier years, there may be more events that go below 4 without getting to that 2 threshold, whereas in later years, almost all the events that are going to go below that 4 milligram per liter threshold are also then going below the 2 milligram per liter threshold, which I think speaks to what appears to be a trend in the intensity of these events. We also wanted to look at how long these events were lasting and how long this area of the bay was in a hypoxic state. So this graph shows cumulative hours across all of the dissolved, the low DO events, all of the hypoxic events in a season. And we noticed what appears to be somewhat of an upward trend in cumulative hours spent below these thresholds, with once again the orange circles being that 4 milligram per liter threshold and the red triangles being that 2 milligram per liter threshold. So why does this matter? As I mentioned at the beginning, hypoxia can have negative impacts on wildlife, and it's especially problematic for benthic populations because they can't get out of the way as easily and can't seek DO-rich waters. And benthic organisms that are intolerant of low DO environments might experience decreased survivorship, where hypoxia is lasting for a few days at a time, as we've seen every year since 2016. So Steve, Craig, and I were able to speak with Dr. Ashley Elgin of NOAA GLERL regarding some of the potential impacts, and she had just returned from completing the 2022 benthic survey that I mentioned on Saginaw Bay. And while her data was not yet compiled, she did share that the team observed fewer Dreissenid mussels in areas of Saginaw Bay that were closer to where this buoy is located, and that the benthic organisms that they did observe tended to be low DO tolerant species like chironomids. And this matters because the relative densities of these different organisms can have impacts across the food web. She also shared a 2018 study that compared Dreissenid presence in hypoxia in the central basin of Lake Erie. And while these are really different bodies of water with different characteristics, it's interesting to think about how low DO events might be a mechanism by which Dreissenid mussels that have a low tolerance for low DO events, it might be a mechanism by which they're controlled. Not that this makes low DO events a benefit, but it is another trade-off to consider in management decisions when seeking to control invasive species like Dreissenid mussels. So now that we know that these low DO events are occurring, we also wanted to get a sense of what are the mechanisms for depletion or recovery of dissolved oxygen. And we looked at a bunch of different things, all of the parameters on the screen now, but this certainly isn't an exhaustive list, and I'll give you the preview that our overall takeaway from this exploration is that a more sophisticated analysis is needed to really understand what's going on. But we did rule out some of the simpler contenders as standalone drivers. So I'll talk briefly about water temperature and an analysis we did on bottom current direction and speed, but know that we looked at a lot of these things. So if you're interested in any of them, I'm happy to talk more about any of them afterwards. So our first thought, and this graph is essentially the same graph that we saw earlier with dissolved oxygen. It's still there in that black line, but we layered on top water temperature, with the light blue being surface water temperature and the dark blue being bottom water temperature. And our thought process on this was based on understandings of stratification in Lake Erie. You know, that's where you see differences between surface water temperature and bottom water temperature associated with drops in dissolved oxygen, and as an indication of stratification. But we also knew that in this area, or Steve and Craig suspected, that there were instances of cold water intrusion into the area where the buoy is moored, that we suspect are coming from Lake Huron and might be carrying dissolved rich water. So we have these two patterns that we were looking for. One where we might see a divergence in water temperatures associated with drops in dissolved oxygen, and then a pattern with the same identifying characteristic, divergence in water temperatures, but associated instead with rises in dissolved oxygen. So we went through each of these years, and I'll show you just one for an example here, where we went through and identified where these patterns were happening in each year. So the green dashed ellipses is indicative of that stratification idea, and the solid red ellipses is associated with the cold water intrusion hypothesis. And then we asked ourselves to look at what might be the things causing this these patterns to emerge in different ways. So that was where we began to explore wave and wind and current. And with the limited amount of time here, I'll just quickly show you what it looked like to look at currents, where the color here is associated with current direction, and then the intensity of that color is associated with current speed. These are current bottom level currents. So we were hoping to see, is there some pattern with the currents that indicates that one of these patterns is more prevalent than the other. And I'll just say that we found that for every example that we found that corroborated our ideas of why that might occur, we found a counterexample, which definitely speaks to just the need for a greater understanding of what's going on here. So I think that brings us to our conclusions from my research this summer, which are basically that episodic hypoxia does occur in at least part of Saginaw Bay and the part where this buoy is located, and that there appears to be somewhat of a trend in cumulative hours spent in a hypoxic state, that might be increasing over these years. And then in some cases, dissolved oxygen appears to rebound with influxes of this cold water, but we still don't know exactly what are the dynamics at play causing those influxes. And then of course, we want more research on how hypoxia might be contributing to reductions or impacts on benthic communities. And then that will help us, hopefully this will help us make decisions on what the management levers are available for contributing to a healthy and thriving Saginaw Bay. So with that, I want to say thanks again to everyone who contributed to this and helped me along the way. And I would love to hear any questions.
Great job, Amy, and very interesting. I haven't seen a lot of data like this from Saginaw Bay. We see a lot of hypoxia data from Lake Erie, so it's really interesting to see that and how it might be increasing over the years. So if anyone has questions, please enter them into the question box, or you're also free to raise your hand and I can unmute you so that you can ask. Okay, question from Anna Baygold. She asks, what do you think is causing the increase in cumulative hours of hypoxia in the Bay?
Yeah, we looked at a lot of different things that might be associated with that. One that is also related to time was lake levels. That was an idea that Craig had when we first saw that graph of cumulative hours, with the idea being that even a meter or half meter more of water might be contributing to might allow for more stratification, which would cause those decreases. And we did bring in lake level data and sort of plot that against those increases, and there seemed to be similar. Lake levels started increasing in 2015, and that's also sort of the beginning of where we saw more increases in episodic hypoxic or intensity of those events and cumulative hours spent below those thresholds. But I wouldn't want to say for sure on any of that. And I'll also note that that the buoy is placed in slightly different locations year to year. So there's some locations where it's at 14 meters and some locations where it's at 12. And those depths did not seem to be related to the intensity of hypoxic events. So while there's a theory behind lake levels being a part of this, I wouldn't want to say strongly that is the answer to the question.
Okay, good answer, Amy. Thank you. Next question is from Abby Hudson, who asks, forgive me if you mentioned this, but is there any in situ hypoxia sampling in Saginaw Bay before the data you have here, potentially from other sources? So the trend seems to be very significant, so more data to verify the trend would be interesting.
That's a great question. And there are other researchers that are looking at hypoxia in Saginaw Bay presently in other locations in the Bay. So I can definitely, if you're interested in learning more, I can follow up with you about that research and send you some links to that. In terms of prior to this, the last sort of big round of research that was done on Saginaw Bay was completed by our very own Craig Stowe as part of the multiple stressors in Saginaw Bay project, and many other researchers at NOAA GLERL were involved in that. And so I would also point you to that research if you'd want to follow up and talk a little bit more. And obviously, Craig Stowe will know a lot more about what all of that entailed.
Thanks, Amy. I'm gonna do one more kind of a combined question. So we've got some questions related to nutrient inputs and their potential impact. So Ed Rutherford asked, are there nutrient concentrations or runoff also increasing over this time period? And related to that, Peter Alsop asked if you looked at any year-to-year differences in tributary inflows or phosphorus loads.
Yeah, those are fantastic questions. We talked at the beginning of my project to see if that's something that we wanted to explore. And from what I remember from those conversations, and obviously Craig and Steve would know more, but from what I remember of those conversations is that there's not necessarily a ton of really great data on tributary flows since in the last 10 years. And that was our metric for whether or not I was going to explore it, was can we map it up with this buoy data? And since there didn't seem to be a time match with that data, we didn't want to go down that rabbit hole. But I can double check in the multiple stressors journal that came out, because I think they did talk a little bit about phosphorus loading. But again, I think that was, if I'm remembering correctly, that was prior to this this chunk of time that I was looking at with the buoy.
Okay, great, very interesting, Amy, and potentially something to look into in the future. And Steve says, excellent presentation, Amy. So thanks very much, Amy.